from the Lawrence Berkeley National Laboratory in Berkeley, California. Eddie has an illustrious career. He's currently the director of the Department of Energy's Joint Genome Institute since 2002, I think, that's right. So his interests are in computational and biological approaches to the development of methods to study genomes, and been involved in the Neanderthal DNA genomics project, and of course his reason for being here is interest in microbial communities. So I won't say any more. I'll just give the floor to Eddie.
Okay, great. It's a pleasure to be here. I've enjoyed getting to listen to the talks and participate in the meeting. The JGI is a large genomic facility operated by the Department of Energy, and usually we have biologists come with problems, and then we help them solve it by providing them enabling technologies as well as analysis capabilities. And so we have sort of frequently naive users come with good biological questions. And so what I'm going to do today is present some recent studies that, and I think it's more biological than most of the talks, and so I'll go through it slowly. And I have about 35 minutes of presentation, so I urge, if there are questions along the way, I suggest you ask them. You don't need to wait to the end, because I don't think it will be packed for time. And so, prior to the DNA era, we had what we would view now as a very simplistic view of life on planet Earth. We had a giant trunk, and two branches came off it. And pretty much what we knew about, or much of what we knew about life, came through observation and maybe microscopy. And then a few miles from here, we entered the DNA era. And it gave us a window into biology that we hadn't had. And we're off to the races. And one of the first things that occurred was that we were cognizant that there was a code. And we called it the canonical code because it appeared that all life had the same vocabulary. And this is one way of depicting the code. There are 4 nucleotides, there are 3 bases and a codon, the smallest unit, and so there are 64 combinations. And this is shown here, the first base G, C, and there's a degeneracy here that leads to codes for alanine. And there are 22 amino acids. And so there's degeneracy. We have 64 possible codons, and we have 22 amino acids and 3 stop codons. And so that was the canonical code. And we view that all of life could communicate via this code. Okay, another thing that came along is that Carl Woese, a very clever quantitative geneticist, began looking at small amounts of data, 16S data, fingerprints of microbes. And from the analysis of this small data set, he came to a very clear conclusion that the tree did not have 2 branches, but rather that there was a 3rd branch, and that there was archaea. And some people, there's some controversy exactly where the branches go off, but clearly there are 3 major domains. And he discovered this with a very small amount of data. And so that's where we're at now. We view this as a canonical code, and we view there are 3 domains of life. But the question is that all? And is there variation within it? And now we have lots of tools that we had, data sets we hadn't had previously. And it opens a window to begin looking for both diversity in the code as well as in the domains of life, and then looking for new discoveries. And so that's what we've kind of been interested in. I'll talk about it in this project. And so if you're going to look for new things, you need to look new places and using new tools. And so the new places is a real focus on uncultured life. And as everyone here realizes that that you filter seawater, only about 1% of it we can grow in the laboratory. Well, 99% we can't, and so therefore that offers us, and what we really know about is that 1% that we can grow. And so there's really enormous potential in that uncultured, and metagenomics provides a window into that. And so we've really been focusing on uncultured, and the purpose of this meeting is to be able to decipher massive sequence data where you don't associate with an organism, you associate it with an environment, and try to derive insights from analysis of that data. And that's a wonderful part about metagenomics, which I really love, is the ability to build biology from sequence data. And then in addition, there's another way to survey the uncultured world is through single-cell genomics, and that hasn't been discussed here very much, but that is a way where you take, and this is really an explosive area in technology development, where you take a single cell, you don't need to grow it, but you can get its genome. And this is in human genetics, it's giant. In looking at uncultured microbes, it's giant. And so just very simply, the way you do single-cell genomics is you go to an environment, you isolate single cells, and you can isolate it by microfluidics, getting individual cells in a chamber. Nothing needs to grow, or you can go in with a pipette, a fine pipette and a very big microscope, and isolate a single cell, or you can sort them, and the variety of approaches to get single cells. And then basically what you do is in this chamber, you lyse the cell, and then you have DNA from a single copy of that genome, and then there are ways, molecular biology ways to amplify it. And it's multiple displacement amplification, and what you use polymerase and use random hexamers that will bind to the DNA, and eventually you build sort of the spaghetti, but you get multiple copies of segments of DNA. And it's been very powerful. You don't get good assemblies from this, but you get fairly good content. And we can begin improving on some of these, but you only have one, you're starting with a single molecule. And so that's another window into the uncultured world. And so the first part of what I'll talk about is we call it microbial dark matter project. And if you have an exploration into uncharted areas, you need a title, and this is our title. And the metaphor is that we've largely spent though we've sequenced thousands of microbial genomes, they've been very small phylogenetic space and mostly cultured organisms. And the goal of this project is really to move away from the lamppost and begin exploring a greater diversity and not depend on only organisms that we can grow in the laboratory. And so this is the landscape that we're looking at. And so what I'm showing you is a phylogeny of bacteria and archaea, and what you're seeing in gray are the branches where we have sequenced genomes. In black are branches that we know exist because we can do 16S amplification, and that all bacteria will have, we think all bacteria have, we can recognize those 16S and amplify them with universal primers, and from them we can get various phyla, but what I'm showing in black are phyla where we just know they exist because there's a 16S that fingerprint that's been associated with them, but we have not a clue about what they do, these organisms. We can't grow them in the laboratory, we don't know their gene content, and so it's really enormous diversity that's out there that we know exists, but we know nothing about them. And so our goal in this project has been to systematically go after these phyla that don't have genomic representatives to. And the approach was to collect samples from around the globe, and we went to sites that we had evidence suggesting that they had these candidates, phyla that we haven't sequenced before, that have been found there, have been seen there in 16S data. And they range from ocean sites to hot vents to thermal vents to geysers. And all these samples were brought back to the JGI, and we had a pipeline for beginning to capture these uncultured organisms. And the way the major technology we use is we would sort individual cells into a tube, And then what we do is we would amplify the single cell, we'd amplify the genome of that single cell that's in the tube. And we did pretty big numbers of this. We had a big pipeline. And then we take a sample from this tube, and then we would sequence the 16S marker in that tube. And the goal was really to go after specifically the branches that we did not have genomic representatives of. And so we weren't sequencing in everything. We were just looking, okay, here's a tube that has this phyla, a 16S marker for this phyla. This is the one we're interested in. And so we chose specifically to sequence those. And we generated about 200 genomes of various quality. And we have various quantitative metrics of the quality of these genomes. They're not very good, but we get a fair sense of gene content from them. And so just to show you how far we got was, here again in red is the newly captured phyla. So we really added an enormous amount of phylogenetic diversity of genomes, which we didn't have previously. And in black are the ones we're still missing. And so we have wanted posters for them. But that's really the goal, is to capture it all. And so pretty much, we really did expand the known, yeah?
What do those triangles mean?
I'm sorry, what?
These triangles, they indicate diversity.
Yes, they indicate particular phyla, right. And the size of them is the diversity within them.
Okay. And that's based on 16S.
Yes. Okay, so I've been involved in lots of genome papers. When you run a genome center, you write genome papers, and they're usually just catalogs, and they're really not that insightful. They don't add that much information. This one was really, the one paper I was involved in, the Neanderthal paper, was really an unusual genome paper because it provided a very deep insight. It told us that we had sex with Neanderthals in the past. And so that was really, that was a genome paper we really learned something from. The original human genome paper, it said that there was an enormous amount of transfer between humans and bacteria. And it turned out to eventually that that was one of the major conclusions in the initial shotgun human genome paper. And it turned out to be just contaminants. And so, so usually genome papers are not that insightful. This one was. There were a lot of insights in it. And the reason is that if you had never sequenced, there was enormous diversity here. And there's all kinds of questions that we could look at in the data about how life works and how the various rules with regard to life, because we had all these organisms very far away in phylogenetic space that hadn't been sampled before. And so there were issues of how the tree of life, how archaea superfamilies that came out, and by now having genomes and be able to compare individual genes rather than just 16S markers. I'll describe a little bit about assigning metagenomic data to organisms, because much of what we do with metagenomic data is you assemble it, you get contigs, but you don't frequently connect it all back to an organism, because we didn't have that many organisms that have been sequenced to be able to connect it to, and this has improved that, and I'll show you that. And finally, there's a whole series of discoveries. And one of the best parts of this paper was, I'm at the Berkeley lab, and it's very much a physics place. And that's depicted in the logo, where you see the big cyclotron, the shape of the building. And so biologists don't get enough respect there. And the nice part about this paper, it had the word, there are a lot of people in Berkeley studying dark matter, unsuccessfully. And this had the word dark matter in the title, so when they search dark matter, they get to learn a little microbiology. And okay, so assigning metagenomic reads to an organism, and so you get a read, but you don't have it in the context of the other capabilities of that organism. And for example, this is a sample that was taken, a metagenomic study that was done on this lake in Canada, and of the reads, you're able to link the reads back to an organism, this blue part of the pie. And so you'd like to be able to link it back to an organism, because there's biology associated with the other genes known to exist in that organism. So but with what we've added in this one study, following the microbial dark matter project, we've really increased the amount of the pie which we're able to link a read back to an organism and be able to associate it with biology. And it's helpful. You begin to understand the capabilities of the microbes in that environment. You thus begin to understand the environment, and biologists can ask questions, use that data to ask questions about it. Okay, with regard to discoveries, we've established rules of how genes are transferred between domains of life. And the question is, are they really rules, or are they just based on our sample, our data set? And so one of the ones is that as far as transfer genes between eukaryotes and archaea, there have been many examples of genes from archaea being transferred to eukaryotes. There are no examples of it going the other way, and so it was felt there was a barrier to the other way. Well, in the study, we have seen clearly eukaryotic genes in the slime mold as an oxidoreductase. We've been able to show, by increasing the diversity of archaea phyla that are sequenced, we've been able to find several phyla that have taken the eukaryotic gene. Okay, so showing that this isn't a barrier to transfer, but rather it's a sampling. So these are the kind of discoveries you can, lots of these occurred in this data set. The one that got my interest was this one, in that two of the organisms we sequenced, GR1 and GR2, had very small genes, 200 base pairs. And so that would say the proteins are, 70, too short. They're peptides. So the average full-length gene is 900 base pairs in length. And so you get a 200 amino acid protein, or 300 amino acid protein. And so these are just fragments of genes. And this comes from the annotation of the sequence. And it was just puzzling. And eventually someone came up with the idea, Natalia Anatova came up with the idea, let's assume that it's not the canonical code. Let's see if there's another code operating here. And she used a code where the UGA stop codon, which normally says an termination, is now coding for an amino acid. And so that allowed the open reading frame, which for the when you're going through and you're annotating, you say there's a stop here, and this is saying no, it's not a stop, but it's rather an amino acid. And so basically, what's going on here is that we've annotated this with the canonical code where UGA is a stop, and it leads to 200 base pair gene predictions. When we assume that the UGA is not a stop, we get greater than 800 base pair, normal sized proteins. And there's other metrics to say that this really is a proper coding region. And we could translate it into a protein, and we could see what UGA was coding for. And it was coding for an amino acid glycine. You can compare it to other known proteins that match it and see that that position was a glycine. And so I'm going to go through this because this is really a central part of what I'm interested in presenting. In that, again, here's the canonical code. There are 64 codons. 61 of them are coding for an amino acid. 3 are for stop. And so here UUU, and these letters indicate a different amino acid. And there are 3 stop codons here, amber, ochre, and opal. And this is the canonical code. The sense codons have tRNAs which recognize them, and the tRNAs are what inserts the amino acid into the growing polypeptide chain. And so the code is translated via the tRNAs, which recognize the antisense of the codon. In addition, the stops are translated via releasing factor. And the releasing factor structurally looks like a tRNA, and it recognizes the codon, but instead of adding an amino acid, it terminates the expanding polypeptide chain. Okay? And when you annotate something, you see a stop codon, you assume that the releasing factor is recognizing it and terminating translation. Okay, well, what's going on in the opal recoded bacteria is that it has decided that this UGA stop codon should not be a stop, and instead should be encoding the amino acid glycine. So it's changed its vocabulary. And it's like driving down the street and suddenly the stop sign, someone's taken away, and It's okay. But one of the crucial things is that the releasing factor normally recognizes that. And so if you want to use it as amino acid, there's a competition between the releasing factor that's viewing it as a stop versus the tRNA that's viewing it as a coding for an amino acid. So what this bacteria did was it got rid of the releasing factor, and it replaced it with a tRNA that inserts a glycine there. And so it's using a different vocabulary. And so, in summary of this project was that we've expanded the genomic representation of the tree of life, and we're on the path to providing an unbiased view of microbial life on the planet as far as we know it. And in addition, it brought up the issue of the universality of the canonical code. Is that the only code that's out there? And so we observed it in 2 of the 200 bacteria that we looked at from uncultured life. And we said, well, let's look more massively at this question by looking at the data set of metagenomic data, which has, and that really gives a window into uncultured life. And so this is the second project, and this is looking at genetic codes operating in the wild. And what we did is we collected all the metagenomic data that we could get. We have a database, RMG, that contains it all. It was close to 15 terabases of data of just reads from different, and more than 1,100 individual sampling sites. And the sampling sites were from, sludge sites, from inside oral cavities, every place where we could collect metagenomic data, we did. And then we developed a strategy for looking for other codes. And in this first pass, the other codes we were specifically looking for are when there was a conversion of a stop codon to an amino acid. And so we were looking for cases where opal, amber, or ochre stop codons were now coding for an amino acid. And the way we looked for them is we read each contig using 4 different codes. One of them was where the canonical code, where we assumed amber, opal, and ochre were stop codons. Then we used a code where we assumed opal was now coding for an amino acid. We used another codon table where we considered ochre was, anyway, and so on. And pictorially, with the canonical code, you look at the sequence, and you run through the sequence, and you predict proteins based on all the normal stop codons. The second code, where we looked at the same thing, except we removed UGA as a stop codon, and we predicted proteins based on that, and continually with each of these. So we used 4 different codes. And so this is summarized here in that we had 15 terabases of data. We assembled that, and we did need assembled data because we really wanted to see multiple places in the same contig of recoding, because we didn't want to just see a single event. We wanted to have a statistic confirming that what we were predicting was really authentic and not just spurious gene finding. And so we demanded that they be greater than KB, and then we put it through the 4 different codon tables predicting.
Eddie,
Yeah.
So in these codon tables, if you assumed that one of these stop codons had been converted to do
Right.
something, you'd have to assume which amino acid it was.
Well, you don't really have to. You just can assume an amino acid. You don't really need to just. Later on, we could decide what the amino acid was, but we didn't need to. We just put amino acid. Yeah. And then there was a statistic that frequently, if you get rid of a stop codon, you will get a bigger prediction. But there are ways to filter what is a real protein and what is a real assemblage. There are statistics to determine the quality of a predicted gene, whether it's just spurious random bases that happen to be coding versus real occurrences that you see. So there is pretty good filtering that you can do. And what we found was that there was tons of recoding in nature. And this is the wonderful thing about metagenomics. It doesn't make assumptions. of an organism. You just can look at these giant data sets, and I think this is what really, as a biologist, really excites me, is an unbiased view of life, just based on sequence data. And so what I'm showing you here is are the different colors represent the different kind of recoding. Orange represents amber recoding, ochre or yellow is ochre recoding, and blue is opal. And the size of the circle is indicating greater than 1%, the biggest size, and the little size is less. And these are showing sites around the planet, as well as around the human body. And you can see that there are multiple instances of recoding, and some environments, 10% is recoded. So it's really, the canonical code is not the rule, or there's lots of examples of recoding. It isn't just a rare occurrence, and most of it is opal recoding in bacteria. And this is showing where we're seeing opal recoding in bacteria. And these are trees, and you can see, and it's not dispersed amongst different phyla. There is an event that occurred, and we can almost begin to time these events, where suddenly this phyla over here per group has a normal code, while everything that descended from here is recoded, where opal is now as an amino acid. And you can see the data over here. Here is the data. It comes from metagenomic data, is that symbol. Single cells are that symbol. And it appears like twice in nature. And this is one we previously knew about. Mycoplasm is an organism we can grow in culture. And we knew that was recoded, where opal was a tryptophan. Here we're saying all the rest, this is the new discoveries, opal is a glycine. And this is supportive that our analysis is valid, because we see true positives, and we know these are known positives, and the new predictions are consistent. They're just within this phyla. One of the questions, why is this occurring? And we do have an explanation, or a partial explanation, is that opal uses it's a UGA, so it's GC rich, and so when bacteria have low GC ratios, they have other choices. They could use ochre, which uses less GC, and they'd rather use the GCs for amino acids. And so as you see organisms, in low GC organisms, they tend to use less opal stop codons, and they use other stop codons. And what happened here is it just reached a point where it decided, hey, I'm no longer using opal as a stop codon. I might as well use it as amino acid. And that's the scenario.
I can imagine that for the individual microbe where this happens, suddenly it would wreck a whole bunch of their genes.
Right, but I think it occurs slowly. It's not a sudden event. Right, and so you can see microbes and so we don't know why microbes have different GC ratios. It's one of the observations. But we see when they have low GC, they have less opal. And there's a clear logic to that, is they'd rather use those GCs because they have another choice of an amino acid, of a stop codon that uses less GC. And so I think it occurs gradually.
So in fact, there would be two different molecules that would recognize.
At one point, there is. Right. And at the end, I'll talk about it, but we, in fact, have worked in the laboratory to synthetically build an organism that doesn't use one of the stop codons, and so we can use it for something else. And nature did it in a slow motion way. We've done it, and other groups have done it using genetic engineering to do it.
One of the assumptions about the GC is it's more stable than the AT binding, so you would expect it at high temperatures, but I think if you look in thermophiles, they tend to have AT rich genes. Right. I have not come to a logical explanation of why. There's different theories about why organisms have low GC, and so I don't know it. But the observation is true. That there are organisms with low GC and they have less opals, and so we don't know the cause of the GC ratio, but the ramifications are organisms
respond to those. That. Go on.
Those three, I'm just trying to figure out which are the clades where you do have. I can't tell which are the clades where you have this.
Okay, I'm sorry. This is really, these are everything below here, over here. These are all in gray are the clades that have opal recoding. Okay, and these are the different labels we give for these subphyla, and these are the sources of the material. And the colors are where we got it. And so, this is fresh water and blue. So there's a lot of information. This is for a figure from paper, and I apologize. There's a lot of information that is poorly described and articulated. Okay. So I'm going to end with a story. and I like this story because I think it really is a three-dimensional biological story that's derived purely from sequence data. And because we can't grow any of these bacteria, but from the sequence data, we can recreate things that are happening. And so one of the things we looked is recoding amongst bacteria, archaea, and phage. In archaea, there's no evidence for recoding. And we have a large data set so that we feel fairly confident that there's no recoding archaea. In bacteria, there's abundant recoding, but it's exclusively opal. And then we isolate, there's more than a terabase of phage data here. And from the phage data, we see that there's a significant amount of opal as well as amber recoding. So they're phage, they're no longer using the amber stop codon. And that's a very curious thing because you'd expect the phage would have to infect a host. And we don't see any evidence of amber hosts. And there's a dogma that's been stated in multiple references that phage need to use the same codon table as the host, that they can't carry the machinery for that. And so we see amber phage, we don't see any amber hosts. And I'll give you the punchline in that, the data says, no, that's not, the dogma is wrong. That there are phage that they don't really care. In fact, they can exploit the differences to damage the host. And I'll take you through the story of this very diabolical phage. And it's a tongue phage. And we've done a clinical study. It's always found in the dorsum of the tongue. We found it in multiple different human subjects. It's 170 kb phage. It's seen in multiple human metagenomic studies, only on the dorsum of the tongue. And we did a study at the JGI where we swabbed people's tongue, and about 10% of the people had, this is our first clinical study, had this phage on the dorsum of their tongue, including myself. And so this is a personal story. And this is a myoviridia phage. It's a phage that infects a host and then lyses the host, a lytic phage. And so it induces the host to make phage genes that sabotage the host, the early genes. And then after they shut down the host machinery, they use the host machinery to make phage lace genes, and then they lyse the host. And so this is the kind of bacteria matches it has. And the clue that the way this phage is operating, and it's infecting an opal recoded host. And the clue to that is that the phage encodes this releasing factor, which is RF2. And let me remind you of the opal recoded host I described earlier on. That has UGAs, and it's using it as an amino acid. And it is figured out to get rid of the releasing factor 2. So it's not competing for stop codons there. Well, the phage infects the host. It begins producing a releasing factor 2. The releasing factor 2 now is recognizing those UGAs as stop codons and truncating all the host proteins. And so it's shutting down the host and then taking over the host, using whatever machinery is already built to translate its own information to lyse the host. And when we see the early RF2 is produced on the early phage genes, and it sabotages translation of the recoded host. And then the late phage genes, there is an amber. Like the late phage genes are amber recoded. They also carry a tRNA that is able to recognize that amber. And so it carries along its own machinery. I don't have an explanation for why we need this amber recoded phage. What the reasoning is, but everything that can happen in biology does happen. So that's the only reasoning that we can see. But anyways, and so what's going on here is we have this host that has rife with opal stop codons, which are now being translated as amino acids, tRNAs, and then we have, and this has no, the host now has no RF2, and then we have this virus that comes in and it has its own RF2, and so early in infection, the virus infects the host. The host is going about its business translating its opal stop codons as amino acids. The phage produces RF2, begins interfering with the host translation, resulting in truncated proteins, and now the phage translates its own, uses its own tRNAs to translate its own amber and lyses the host, and lyses the host, and the game's over. Okay, other evidence for this is also gotten from the metagenomic data is the use of CRISPR-Cas system. And who here has a familiarity with the CRISPR-Cas system? Okay, it's really a fantastic discovery. Who's heard of Cas9? Okay, these are really revolutionary advances in molecular biology that's occurred in the last year and a half, and you will hear about it. It is really it's like restriction enzymes. It's one of the most profound discoveries in molecular biology in the last 20 years. And what it is that when phage infects hosts, sometimes the host has its own immune system, and that's what this is. Like when we're infected with a virus or a protein, we develop an immune response to it, and we have a memory that tells us that we've seen this, and we can respond to it when we see it the second time. The phage, this system's the same thing. It's a memory system for for the host to recognize phage that have infected. And what happens is that sometimes there's this warfare going on between the host and the phage, and in some cases the phage will infect a host and produce a viral DNA, but then that DNA will be recognized by the host, and it'll break down that DNA and integrate it in its genome. Okay, and these are called CRISPRs. And what it is that in many bacterial genomes, you'll see little bits of phage in their genome. And eventually, if the host has developed a system where if it gets infected, so this host survived this infection by this phage, and all the offspring of this host contain a bit of its memory of the phage sequence. And if the phage comes again, it will be able to fight it by having that memory. And so what we have is that when phage affects host, it leaves a footprint that it's been there. And so what we did is we searched all the CRISPR sequences from the 15 terabases of metagenomic data, looking for footprints of that amber phage that have been there. And what we did is that we found in this multiple examples that the amber phage sequences were in these CRISPR sequences, saying that they were there. And then we could see that it was surrounded by opal recoded bacterial genes. And so though we didn't see this event, this is another level of evidence from metagenomic data that this occurred. And we see genes with a canonical code. And so in summary, the dogma that phage must share the same genetic code as a host bacteria is not true. In fact, phage can exploit the differences. And this is the kind of thing you can only get from metagenomic data. And it says there's an abundance of recoded microbes in the environment, and that the canonical code is not the rule. It's not the exception, it's the dominant code being used, but there are many other variations of it. And it has some relevance in that there's a great effort to build synthetic organisms. And one of the real aims is to make ones that there's a firewall where you build a synthetic organism, it will use a different code, and therefore can't be infected by phage and can't spread its DNA into the environment, because the canonical code was felt to be universal, and it would be a barrier to DNA that had a bunch of stop codons as amino acids recognized by other organisms would not be properly translated. But this study says no, that viruses know how to overcome these differences and in fact exploit them, and it says there are lots of organisms out there that have different codes. And so, okay, so that's the first phase of the microbial dark matter project, and it was really looking for variation in known phyla. We were going to 16S's and looking for organisms that hadn't been explored. The next phase is really to try to look for new things, entirely new things. And so the study has looking at lots of different sites. The real goal here is to see if we can find new domains of life. And we spent a lot of time, looking for signals in space for other life. My money is that we're going to find it on Earth, and I think metagenomics is the window into that. And so this study is really aimed at that. We're looking at many different environments, and we're not demanding that the cells grow, but there is something to getting, and so we're scaling our ability to do single cells. And so being able to look at 10,000 and a million cells, and being able to do 16S identification, but look at everything that doesn't have a 16S marker. With the hope that some of these will be very novel. And this information is probably in metagenomic data, but it's very hard to link it together. Like, so much metagenomic data does not track back to an organism, and you just assume it's somewhere connected to an organism. But here you're able to get get more content of a particular organism, defined as an organism. And so the question is, are there other domains? And if you're looking for exercises, to spend your next 3 weeks, this is a noble project. Finding new life is important. And so I'll end there, and I do want to acknowledge a series of talented people drove this project: Tanya Woking, Nikos Kripidas, Natalia Ivanova, a couple of postdocs, Chris Rinking, Patrick Schwenke, and Alex Zerba, who's going to give a talk later today. And so, thank you.
Thank you, Eddie. That was a tremendously exciting presentation. I don't think anybody could fail to be excited. So we've had some questions. Let's have some more. Yes.
Eddie, have you gone back in and recoded some of the sequence genomes from some of the other molecules, the tenoracutes, to see whether or not many of the other, say, the phytoplasma, the spiroplasma?
Well, but the thing is, and so they annotate well, and so it'd be hard to predict that they would have recoding as far as stop codons. But there's, we have other strategies. We're going to now look at recoding the amino acids, but it'd be hard to imagine that you'd get better predictions with, it stands out when you have stop codon recoding, and so and so we haven't done that, but I don't think that would uncover anything.
Does this affect the gene calling in the pipelines that are used in IMGM? Are you using things that are based on homology and can deal with potential things that look like a frame shift then, but are not?
Right. No, I think that is the case. Yeah.
About the first part of the talk, what can you say about the relative abundance of the microbes that you found by single-cell genomics?
Yeah.
Where are they?
Yeah. I think that's really based on 16S data. And so it's, I think they're highly abundant and non-abundant. I think the ability of an organism to grow in culture is just if we did culturing different ways, we'd have a different set of organisms that could grow in culture. So I think we're not looking at any unique features of the new organisms that we've identified. They're not abundant or not. They must be pretty abundant because we were able to pick them out of, the 3,000 cells that we looked at. So. But this is most of what's out there, you know, because if you take, you know, many samples, only a small fraction will grow in culture. So it's probably more, you know, so it is a major component. Yes.
I've got a question related to the stop codon again.
Yeah.
Is it possible that a given bacterium is using this codon as a stop codon
Right.
in both ways, and also as an
amino acid
codon?
Yeah. Yeah. I mean, there are examples where there's read-through of stop codons, and so and so there are examples where that occurs, but usually
Opal or
what?
But
not in these. There's that these organisms, because you don't get a full-length peptide unless you assume the stop codon is coding for an amino acid. And so I think there are organisms that will do both, but I don't think it's absolute. Like once they decide that they're bisexual organisms, they use it both ways. But once they decide they're going to go opal, they can't go the other way, you know, entirely opal.
So I'm just wondering whether the new genomes that you produce, are they in GenBank, or do we have to go?
Yeah, no, they're IMG as well as GenBank. They're all deposited.
So I have a question. So you'd be looking for these sort of new things.
Right.
You had a fourth branch of the tree of life.
Right.
And you don't quite know what you're looking for. So I guess you're really just looking for in the first place, sort of statistical
Yes.
anomalies.
Yes, no, that's exactly it. And I think there is a, you know, I'm very interested in Neanderthals. And it is amazing, because it's quite similar to this, because they're not coming back, and we're not going to have a time machine. But from sequencing, we really are learning an enormous amount. We're learning about their history with humans. And so it's a little parallel, and like we're learning that Neanderthals clearly mated with humans as humans moved out of Africa. And so we see Neanderthal sequences in Europeans and Asians. We don't see it in African sequences. And so therefore we know in the genomes, we can learn about Neanderthals from looking at our genomes. But also, we're learning that we mated with other hominids. Because you see in our genomes, we see these chunks of DNA that don't match Neanderthals and don't match people in Africa. And we can tell that those are recently, that they're big chunks of DNA with a lot of variation that are shared amongst Europeans and some Asians that does not match the Neanderthal genome, and we do not see it in Africans. And so therefore it's telling that we mated with other hominids after we left Africa. And that we don't have a clue who they are. And so in some ways, it's a little parallel to this. You look at this data, and they came up with clever ways of so there are statistical ways to begin looking for novelty. And that's the gauntlet I'll throw down for you, because we're very interested in that, and we're going to be generating data along those lines. And I think that Carl Woese had a very small amount of data, and it was standing in front of everyone, and he was able to identify another domain. And we now have lots of data. And so it's really, the challenge is to be able to find out ways to sift through it to identify.
I mean, it's not just about stop codons.
Right.
Anything.
Right.
You mentioned that you had some samples where there's no 16S.
Right.
Have you looked at any of
No, we haven't, but that's the next study. That's the goal, is to do that.
That'd be really interesting.
Yeah, that's the goal, is to do that.
Yeah. The sort of paradigm for a bacterial genome is a single circular genome,
Right.
maybe plasmids, maybe
Right.
And subsequently, the linear chromosomes, there are organisms that have two chromosomes, maybe one circular, one linear,
Right.
and so on. Has any of that shown up in these novel organisms? I don't know if you can actually...
Because you can't grow them, you can't see them.
No, I know that. Sorry, you can't grow them, but you're sequencing them. Does the type of data...
We get really crummy data from the MDA, and so...
So you can't say anything about
Yeah,
it?
can't say anything about them.
Okay.
I think they're no different. I think they're just, we just don't have the culture conditions right, and they're nothing special about them. But anyway,
So, I mean, you have the opportunity now, if you scale up, to sequence essentially clone mates. of the cell.
Right, it's a way to get better assemblies.
Where to get that.
Right, and we do that. We do that to improve, our assemblies of uncultured organisms.
So you may have nearly completed genomes.
Yes, we do. In fact, yes, right. There's nothing, I don't think there's anything special about these uncultured, just that their metabolic requirements we haven't matched. And if you had a postdoc that wanted to spend, or a graduate student wanted to spend 5 years, they could probably figure out how to grow them. And in fact, from having the sequence, people have gotten better. From knowing the sequence, you can figure out what the requirements are. And so there's nothing special about them. I think we've just opened phylogenetic space using single-cell sequencing, but I don't think there's anything magical about it.
You were saying that it was difficult to do assembly for single cells. Can you give us something of insight about what it is that is the challenge there?
Alex Zerba,
I would talk about this later.
he's going to talk about this.
I'll give you some
You're
examples.
punting.
I am punting.
Okay.
Yeah.
So I have another question. I think it was your first or second slide, and you sort of disproved this dogma that was it phages only infect
Right.
eukaryotes?
Right.
The other way.
Right. No. Okay. Well, there are two dogmas. One was gene transfer between archaea from eukaryotes to archaea.
Yes. That's
Right.
Yeah.
Right.
Yeah, okay. So now with these altered varieties of stop codon, presumably that would mean that an organism that
Yes.
I mean, there's some
It would inhibit lateral transfer.
Absolutely.
Right. And so you have an organism that has and that's what they assumed when they, as I said at the end, we're trying to build these synthetic organisms that have different code. The hope is that it would not be able to transfer its genes to environmental organisms that have a normal code. And so, and so you would expect less transfer between these. And we haven't really been able to look at that, but I pondered it.
Yeah.
Yeah, no, that's a major point.
You've been looking at your data set. I mean, have you been looking for instances where there are only 10 of the amino acids, for example, ever occur, reduced codon sets?
We are beginning. Yes, and so there are organisms that these are great questions. These are important issues, and your GC ratio really affects which because you reduced your work where you reduced the amino acids to 4 categories. Well, if you have GC ratios, you decide which ones to use because their codon usage of GC, and so so you can look in the data, and I'm sure as GC ratios decrease, you decrease your codon. And I don't know if people have looked at it, but I'm sure it's going to be the case where people are going to choose similar amino acids that have the better GC ratio for low GC as well as high GC. Yeah.
Just a comment. It seems to me that if at some point over a period of time there's a transition between the interpretation of a stop codon within an organism, it wouldn't make sense if it suddenly developed this extra tRNA that could sort of interpret otherwise what would be a stop codon to something else, because you then got kind of an ambiguity.
Yeah.
But it seems more logical to me that if you're in an environment where you have to manage with a low GC content, you're probably just going to drop one of the stop codons.
Right.
And then you manage with just 2.
No, but they managed with 2, but they recoded it.
Exactly. So
Right.
that would be the logical sequence of events.
Right.
Where you completely drop one stop codon, then it's free to be used.
Right. Exactly. Exactly.
That seems to be the logical way.
Yeah. I mean, it's very yeah.
Okay, do we have any further questions? Oh, great. Thank you very much.
Thank you.
Eddie. That's